right, so one more reminder, if you lost your Metro card, please see me. I've got it in my pocket. But our next speaker is Wendy Goodson at the Air Force Research Laboratory. And Wendy, come on up and I'll...
Thank you and good afternoon. I especially want to thank Paula and Lynn for the opportunity to present today. It's been a wonderful couple of days. Paula was really excited to invite us to this meeting to expose us to the diversity of types of MoBE projects that were going on that were funded by the foundation. And I have to say, yeah, it's opened my mind in terms of problem solving for these various environments and problems. My collaborator, Brad Stevenson, and I like to say that usually at a conference we're the special snowflakes in the room, and it's nice to be among so many other special snowflakes today. So I am a senior research biologist at the Air Force Research Laboratory, and I hope to give some perspective about some of the work that we're doing on microbiomes of military aircraft and their connections to biocorrosion, in particular to polymer biodeterioration. I do want to start by thanking my team. I have a fabulous team in the Materials and Manufacturing Directorate at AFRL. Two of them are here today, Vanessa J and Audra Crouch, sitting in the back row back there. Also, we work with a great team of collaborators at the University of Oklahoma. Brad Stevenson is here, and Blake Stamps is here too. He's moved on to John Spears Lab at Colorado School of Mines, but he's a recent grantee from the Sloan Foundation for International Space Station Microbiome, and it's the impact of that microbiome on material degradation. And so we look forward to working and working together in the future and passing on some of the lessons learned. So I want to start out with this chart. This is a riff on a chart that Ed put up the other night, and encouraging us about looking at the characters in a system, and then the causality of those characters. So what they're doing within a system, what kind of functions that are happening in that system. And, you know, I don't mean this talk in any way to be complaining, but it's complicated. And I'd like to just pass on how it's complicated for us and some of the challenges that we're going through and how we're trying to solve those challenges. So here's why we're in this business. Military aircraft can be excellent reservoirs for microbiology, and really there are two impacts there. There's a crew health implication and there's a material biodeterioration implication. This picture here on the left-hand side of the slide is a compartment on a C-5, one of our Gigongo cargo aircraft. That compartment is the width of my shoulders, and it's about 6 feet deep. And the hydraulic lines for the nose landing gear run through this compartment, and they leak and break from time to time. And you can imagine the surprise and maybe horror of somebody as they crack open that compartment, has to go into that compartment when there's that level of microbiology in there. Literally, you are nose to nose with a whole lot of fungi. So that's one issue. Another issue is if you crawl into that compartment and stare hard at the polyurethane top coat that's in there, you can see that the fungi are degrading that top coat. It's been stated that the annual corrosion bill for the DoD, Department of Defense, is $5.4 billion. So $5.4 billion of taxpayer dollars go to feeding the corrosion machine, and it's also been estimated that 10% of that corrosion bill is due to microbial corrosion in systems. I don't know if that number is true or not. That number actually is derived, that 10% is derived from the oil and gas industry. I would say we haven't shown that for certain within military systems, but nevertheless, you take 10% or you take 1% of 5.4 billion and it's still a pretty big number. And certainly we've seen it with our own eyes about the impacts on our systems. So my lab has spent the last 6 years looking at fundamental studies of polyurethane deterioration in our laboratory. So we've looked at things like the impacts of coating chemistry on this process. We've isolated some of the hydrolases that are involved in this degradation process. And we pay a lot of attention to physiology. So when you vary things like temperature and humidity within a system, what that does to the ability of a particular microorganism to degrade a coating. But you can only get so far with lab studies. At some point, you have to go out into the real world if you want to make an impact in that real world and determine what you can do there. And so what we'd really like to do is to be able to assess risk and determine causality in some of our in our aircraft. And so I'd love to go into a typical aircraft and swab that aircraft and then basically give them a risk analysis for the potential of the microbiome of that aircraft to cause corrosion within that system. And I'd also like to someday be able to have a maintainer come up to me with 2 cans of paint and say, which one should I use on this aircraft? To be able to give those folks a way to test these different coatings in a relevant way so they can predict what the lifetime of those coatings would be on our systems. In order to do that, you really have to think about 3 different pieces of the puzzle in the environment. First of all, the types of microorganisms are there. That gives you a sense of their genetic and biochemical potential. Really importantly, you have to think of microorganism abundance. They need to have the critical mass present in order to make the biochemistry matter, essentially. And finally, there's this microbial physiology and its interaction with the environment, this codependence of these 2 things, which will further drive this degradation process. And so rather than looking at severely contaminated systems, we wanted to establish a baseline of what types of microbiology was present on our aircraft. And so we went out and started sampling cargo aircraft. And we started with 5 of them. We sampled various areas throughout the aircraft, floors, walls, dry bays, that's a maintenance area, flight controls, exit entry points, and the urinal. We looked at suspected contaminated areas as well as areas that looked pretty clean. And we sampled frequent human contact points. And you can see here there's a list of materials that we targeted across all of these aircraft. What we did was go in with swabs, and we did 2 things with those swabs. We cultivated the microorganisms, actually Audra cultivated the microorganisms, both on TSA, tryptic soy agar, and potato dextrose agar media, not only to understand who was there, but also to be able to count the number of microorganisms there. And I'll tell you, I know cultivation-based approaches aren't always the favorite thing for this community, but they're critically important for us, and I'll explain why in a minute. We also took a subset of our samples to do community analysis by 16S and 18S ribosomal DNA sequencing, and we also did qPCR. So through a combination of these techniques, we get an idea for who's there, who's been there, and how many of them are there. So one of the first questions we asked was, do certain locations on aircraft harbor microbiology? So we looked, in this case, I'm showing you the cultivation data, the log CFU per centimeter squared in fungi and bacteria based on the types of plating methods that we used here. We do get similar results when we look at copy number. When you try to roll up all the aircraft into these bars versus locations, you can see there are really no trends. There are no particular hotspots for fungi on these aircraft, although some spots initially look a little bit more contaminated than others. But you can see, especially when you compare the fungi to bacteria, that there are no particular hotspots, and it's certainly not consistent between the fungi and the bacteria. So the other thing that you can see here is that generally numbers of cultivable bacteria are greater than those for fungi. Again, similar results with copy number. So we also thought, well, if we don't have hotspots on aircraft, do we have hot aircraft? And there seems to be some evidence for this. So for instance, aircraft number 1 here, you can see has quite a bit more microbiology on it, both for fungi and bacteria, than the other two here. And you can extend this for all 5 aircraft that we looked at. And there are different levels of colonization here. It turns out this aircraft actually had a lot of standing water in it. There was so much condensation on the roof of this aircraft in the interior that it was almost raining inside the aircraft when we sampled it. So there was standing water on the cargo bay floor as well as underneath the cargo bay floor, which surely helps the microbiology to flourish. So one of the questions we'd love to answer, and we're not quite there yet, but when you see high levels of microbiology, is this evidence of an outbreak? And this is when it becomes really important to consider abundance of bacteria here. And so what we did was to plot face phylogenetic diversity index versus copy number. And you can see here for the bacteria in particular that when you have a really low copy number, this index is all over the place. So basically this makes sense that what we're sampling here is basically background. It's just stuff that's falling on a surface that's not necessarily growing on the surface. And when you start to see higher and higher copy numbers and you start to see growth, now you see an effect on the diversity there. So we see less and less diversity as you get higher copy numbers. So this is something that we're interested in looking at more. We also wanted to look at whether there were similarities in the communities based on where we were sampling from. So we binned all of our sampling locations into either the control, which is basically a background reaction. We have the dry bay here. We have all the floor samples. We have human-associated samples, which includes controls and handles. We have other inside areas like the walls and the bulkhead. And then we compare that to what we get when we sample the outside of the aircraft. And what you can see here is that, you know, there's a whole lot of clustering. There's not a lot of separation in the samples here. But again, we want to consider copy number when we look at this. And so if we change our color scheme to look at copy number, now all the blue here and the green are the lowest copy numbers by 16S and 18S. And so what we're seeing here is basically background amplification. So this is really low levels of microbiology that we're amplifying. We're amplifying the background. And so then when you start to pay attention to these higher copy number samples out here and here, and then compare that back to the original data, now we're starting to see that you can actually compare communities here in a relevant way. You require that diversity with that high copy number in order to start to see this. And now we can start to see that the outside of the aircraft looks a lot like the floor of the aircraft, and that makes perfect sense because these cargo aircraft, they have a ramp, they're driving equipment on there, there are people that are tramping all over the inside of this aircraft. The outside of the aircraft does look like the inside of the aircraft as far as microbiology goes. So we wanted to ask who's there. So here's a taxa summary by location. And again, I'm going to highlight the locations where we have high copy number because we want to pay attention to those locations as being bona fide communities as opposed to just background. And so I'm just going to highlight those in particular. And what you see here is that we've got basically a lot of alpha, beta, and gamma proteobacteria that represent kind of your quintessential environmental microbiology, the heterotrophs and oligotrophs of the world that are just really good and really hardy and good at living in a lot of different environments. So that's not entirely unexpected. We also wanted to look at where these organisms on the aircraft might have come from. And I have to say, Brad and I went back and forth a lot on this as to whether or not we could actually do this analysis, and it's still a work in progress here. So here are our aircraft samples that are standing out by themselves in the middle here, as opposed to home or office or skin microbiomes. And of course, you're probably sitting there saying, what about something like soil and air? And we really struggled with coming up with good libraries to do this comparison because you know, these aircraft fly all over the world. They're in contact with all kinds of different types of soil. where do you start with in terms of comparing what type of soil microbiome to these aircraft? And so we're still kind of doing surveys of different soil libraries that are available in order to make more sense of this source tracker information. Okay, so I mentioned at the beginning the importance of doing the cultivation studies. And one of the reasons why we do those cultivation studies is that we want to be able to screen our isolates for their ability to degrade polyurethane. And so Audra isolated 1,300 isolates from these systems and tested every one of them in this polyurethane degradation assay. So what this assay is basically, this is a plate of colloidal polyurethane, and you spot a colony on top of that polyurethane, and if it secretes an esterase or a hydrolase or a lipase, it will form a zone of clearing around the edges of that colony, and that will be a positive hit for us. So out of 1,336 isolates, we came up with 214 polyurethane degraders. So it's important to understand how biased your study is. We know that cultivation is biased inherently based on the type of media that you're selecting. And so we wanted to think about that too. So this is a plot that shows you in the black out here is all the sequences that we identified through molecular analyses. And then the red highlights here are the ones that we cultivated. So clearly, again, not surprisingly, we didn't cultivate everybody in the system. So we know that when we're looking at polyurethane degrading organisms, we're only starting with a subset of what's in there. However, with that caveat in mind, we went ahead and looked at different types of fungi that we had in our collection and different types of bacteria that we had in our collection, and looked at the total number of isolates and how many, what the proportion of those isolates, what those strains were in their ability to degrade polyurethane. And so when you start to look at this, you start to realize that the fungi look like pretty good polyurethane degraders, especially compared to the bacteria. And so we have looked at these one by one in our lab too, and without a doubt, these fungi are often much better degraders for whatever reason. Either the enzymes that they're making are much more catalytically active, or they're secreting more enzyme, but they're just better degraders than the bacteria are. We also can look at this a different way, where we put these are all the cultivars on the outside of this circle. On the inside are all the ones that are polyurethane degraders. And so again, you highlight this side is the fungi, this side is the bacteria. You can see how many fungal degraders there are as compared to bacterial degraders. And so then I want to go back to the concept of cell number and what that really means, because way back in the beginning I told you that when you count CFUs, there seem to be more bacteria than there are fungi, but now what I'm saying is that the fungi are actually better degraders than the bacteria are when we look at them on a case-by-case basis in the lab. And so, yeah, you may have fewer fungi by CFU or copy number, but they actually may be more impactful to the system. And then we start to think about other issues that we have here, and basically you got to think about numbers versus body plan. So, you know, what's the impact of 10 fungi that are 10 microns big versus 10 bacteria that are 1 micron big? So, you know, we've got a surface to volume issue here, we've got an aspect ratio issue here, and especially when you consider the fact that these fungi often are forming hyphae, which then are increasing their volume or their coverage by an order of magnitude, then you start to see how these fungi can be really potent degraders within our systems. So with that, within the Department of Defense, we're fond of giving ourselves grades, coming up with report cards for how we're doing on certain things. And so here's my report card of the day for where we are in really trying to establish this degradation index for aircraft. So basically, who's there is something that we can do relatively easily, I would say. The how many, as I said, depends on how you ask the question. If you're talking about cultivation versus copy number versus body plan, those are three different answers that you'd get. course, there's also the question that we've heard over the last 2 days of who's alive and metabolically active, and that's really difficult for us to answer right now. We've played with PMA assays in the laboratory, but that's not the easiest thing to do for environmental fungi, which were built to withstand the test of temperature and changing conditions. And those PMA assays work great for wussy lab bacteria, but not great when you're dealing with fungal cells. The what doesn't matter, still difficult for us. We can't ignore the physiology of these systems. When we have a separate study that we do in fuel systems, and we have done some in situ studies of biocorrosion in the presence of biomass, where we can get real-time measurements of both of corrosion and biomass, but on these aircraft, it's a lot harder thing to do. Currently, Vanessa is optimizing some parameters to do a metatranscriptomic study so we can use a cultivation unbiased approach to try and pull out some of the potential degraders out of the system. So grabbing the RNA and doing a functional genomics study to see if we can pull out some active degraders that we might not have been able to cultivate the first time around. So with that, I'll say thank you, and if there's time for questions, I'm happy to take some.
Questions, Wendy? We have time for 1 or 2 questions.
Hello, AAA assigned policy fellow. Great talk. Part of my PhD actually was studying the physiology of bacteria and biofuels. And my question to you is, have you guys considered trying to understand the electrochemical gradients of your isolates to see how that might contribute to the degradation of polyurethanes?
Did you say electrochemical gradient?
Yes. Membrane gradient.
Oh, membrane gradient.
Yeah.
No, actually, no, we haven't really considered that very much. I'd be happy to chat about it afterwards.
Thank you so much.
Yep.